Hello everyone. In our previous video, we discussed about the dynamic nature of the plasma membrane. Now we will move to the next topic, which is the movements of the substances across the cell membranes. So, because the contents of cell membranes are completely surrounded by its plasma membrane, all communication between the cell and the extracellular medium must be mediated by this structure. In a sense, the plasma membrane has a dual function. It must retain the dissolved materials of the cell so that they do not simply leak out into the environment, while on the other hand, it must allow the necessary change of materials into and out of the cell. The lipid bilayer of the membrane is ideally suited to prevent the loss of charged and polar solutes from a cell. Consequently, some special provision must be made to allow the movement of nutrients, ions, waste products, and other compounds in and out of the cell. There are basically two means for the movement of substance through a membrane, possibly by diffusion or actively by an energy-coupled transport process. Both types of movements lead to the net flux of particular ion or compound. Net flux indicates that the movement of the substance in the cell which is in flux and out of the cell which is efflux is not balanced, but that one exceeds the other. Several different processes are known by which substances move across membranes: simple diffusion through the lipid bilayer, simple diffusion through an aqueous protein-lined channel, diffusion that is facilitated by protein transporter, and active transport which requires an energy-driven protein pump capable. The energetics of solute movement. Diffusion is a spontaneous process in which a substance moves from a region of high concentration to a region of low concentration, eventually eliminating the concentration difference between the two regions. So diffusion depends on the random thermal motion of solids and is an exergonic process driven by increase in entropy. But we will restrict the following discussion to the diffusion of substance across membranes. So this figure shows the four basic mechanisms by which solute molecules move across membranes. The relative size of the letters indicates the direction of concentration gradients. So in simple diffusion through the bilayer which allows proceeds from high to low concentration. And then B is the simple diffusion through an aqueous channel formed within an integral membrane protein or a cluster of such protein. So movement is always down a concentration gradient. So in the C, so as you can see, letter C facilitated diffusion in which solute molecules bind specifically to a membrane protein carrier. And then the last one is the letter D, which is the active transport by means of a protein transporter with a specific binding site that undergoes a change in affinity driven with energy release by an exergonic process such as ATP hydrolysis. So in here, movement occurs against a concentration gradient. So the figure below shows the examples of each type of mechanism as it occurs in the membrane of an erythrocyte. Diffusion of substance through membranes. Two qualifications must be met before a non-electrolyte can diffuse possibly across a plasma membrane. The substance must be present at higher concentration on one side of the membrane than the other, and the membrane must be permeable to the substance. A membrane may be permeable to a given solute either because that solute can pass directly through the lipid bilayer or because the solute can traverse an aqueous pore that spans the membrane and prevents the solute from coming into contact with the lipid molecules of the bilayer. Discussion of simple diffusion lead us to consider the polarity of a solute. One simple measure of polarity of a substance is its partition coefficient, which is the ratio of its solubility in a non-porous solvent, such as octanol or vegetable oil, to that in water under conditions where the non-porous solvent and water are mixed together. This illustration shows the relationship between partition coefficient and membrane permeability of a variety of chemicals and drugs. It is evident that greater the liquid solubility, the faster the penetration. Membranes are highly permeable to small inorganic molecules such as oxygen, carbon dioxide, nitric oxide, and water, which are thought to slip between adjacent phospholipids. In contrast, larger polar molecules such as sugars, amino acids, and phosphorylated intermediates exhibit poor membrane penetrability. As a result, the lipid bilayer of the plasma membrane provides an effective barrier that keeps these essential metabolites from diffusing out of the cell. Some of these molecules, like sugars and amino acids, must enter cells from the bloodstream, but they cannot do so by simply diffusion. Instead, special mechanisms must be available to mediate their penetration through the plasma membrane. The diffusion of water through membranes. So, water molecules move much more rapidly through a cell membrane than do dissolved ions or small polar organic solutes, which are essentially non-penetrating. Because of this difference in the penetrability of water versus solutes, membranes are said to be semi-permeable. Water moves readily through a semi-permeable membrane from a region of lower solute concentration to a region of higher solute concentration. This process is called osmosis, and it is readily demonstrated by placing a cell into a solution containing a non-penetrating solute at a concentration different than that present within the cell itself. When two compartments of different solute concentration are separated by semi-permeable membrane, the compartment of higher solute concentration is said to be hypertonic, relative to the compartment of lower solute concentration, which is described as being hypotonic. When cell is placed into a hypertonic solution, the cell rapidly gains water by osmosis and swell as illustrated in the figure A. Conversely, a cell placed into a hypertonic solution rapidly loses water by osmosis and shrink, as what is illustrated in the figure B. This simple observation shows that a cell's volume is controlled by the difference between the solute concentration inside the cell and that in the extracellular medium. The swelling and shrinking of cell is slightly hypotonic and hypertonic media are usually only temporary events. Within a few minutes, the cells recover and return to their original volume. In a hypotonic medium, recovery occurs as the cell loses ions, thereby reducing their internal osmotic pressure. While in a hypertonic medium, recovery occurs as the cell gains ions from the medium. So once the internal solute concentration, which includes a high concentration of dissolved protein, is equal to the external solute concentration, the internal and external fluids are isotonic, and no net movement of water into or out of the cell, as what you can see in the illustration and the figure C. Osmosis is an important factor in multitude of bodily function. So your digestive tract, for example, secretes several liters of fluid daily, which is reabsorbed osmotically by the cells that line your intestine. If this fluid weren't reabsorbed, as happens in cases of extreme diarrhea, you would face the prospect of rapid dehydration. So plants utilize osmosis in different ways. Unlike animal cells, which are generally isotonic with the medium in which they are treated, plant cells are generally hypertonic compared to their fluid environment. As a result, there is a tendency for water to enter the cell, causing it to develop a trigger pressure that pushes against its surrounding wall, as illustrated in the figure A. So trigger pressure provides support for non-woody plants and for the non-woody parts of plants, such as the leaves. If a plant cell is placed into a hypertonic medium, its volume shrinks as the plasma membrane pulls away from the surrounding cell wall. A process is called plasmolysis, as illustrated in figure B. The loss of water due to plasmolysis causes plants to lose their support and wilt. In 1992, Peter Agra and colleagues at Johns Hopkins University identified a class of integral proteins called aquaporin that allow the passive movement of water from one side of the plasma membrane to the other. So each aquaporin subunit contains a central channel that is lined primarily by hydrophobic amino acid residua and is highly specific for water molecules. So billions of water molecules moving in a single file can pass through each channel every second. Now is the diffusion of ions through membranes. The lipid bilayer that constitutes the core of biological membranes is highly impermeable to charged substance including small ions such as sodium ion, potassium ion, and calcium ions. Yet the rapid movement of these ions across membranes plays a critical role in a multitude of cellular activities, including formation and propagation of a nerve impulse. secretion of substances in the extracellular space, muscle contraction, regulation of cell volume, and the opening of stomatophores on plant leaves. As might be predicted, mutation in the genes encoding ion channels can lead to many serious diseases. In 1955, Alan Hodgkin and Richard Keynes of Cambridge University first proposed that cell membranes contain ion channels, that is, openings in the membrane that are permeable to specific ions. In late 1960s and 1970s, Bertil Hale of the University of Washington and Clay Armstrong of the University of Pennsylvania to obtain evidence for the existence of such channels. And in late 1970s and early 1980s, the final proof emerged through the work of Bert Sapman and Erwin Herr at the Max Planck Institute. They developed techniques to monitor the ionic current passing through a single ion channel. This is accomplished using fine micropipette electrodes made of polished glass that are placed on the outer cell surface and sealed to the membrane by suction, as seen in this picture. The voltage across the membrane can be maintained at any particular value, and the current originating in the small patch of membrane surrounded by the peptide can be measured. So this studies marks the first successful investigation into the activities of individual protein molecules. Today, biologists have identified a variety of ion channels, each formed by integral membrane proteins that enclose a central aqueous spore. So most of the ion channels that have been identified can exist in either an open or a closed conformation. Such as channels are said to be gated. The opening and closing of the gates are subject to complex physiologic regulation and can be induced by a variety of factors depending on the particular channel. So two major categories of gated channels are: Number 1, voltage-gated channels, whose conformational state depends on the difference in ionic charge on the two sides of the membrane. And 2 is sterile gate channels, whose conformational state depends on the binding of specific molecule, which is usually not the solute that passes through the channel. Some ligand-gated channels are open following the binding of a molecule to the outer surface of the channel. Others are open following the binding of a ligand to the inner surface of the channel. For example, is the neurotransmitters such as acetylcholine act on the outer surface of a certain cation channels, while cyclic nucleotides act on the inner surface of a certain calcium ion channels. In 1998, Roderick Buchanan and his colleagues at Rockefeller University provided the first atomic resolution image of an ion channel protein, in this case, a bacterial potassium ion channel called KCSA. This landmark has been followed by a series of investigations that has revealed the mechanism by which these remarkable molecular machines are able to select overwhelmingly for potassium ions over sodium ions, yet at the same time allow one and through rapid conductance of potassium ions through the membrane. The KCSA channel consists of four subunits, two of which are shown in the figure. Each subunit in the figure is seen to contain two membrane-spanning helices, the M1 and the M2 helix, and the pore region at the extracellular end of the channel. P consists of a short pore helix that extend approximately one-third the width of the channel and non-helical loop, which is the ones colored in brown, that forms the lining of a narrow selectivity filter, so named because of its role in allowing only the passage of potassium ions. The lining of the selectivity filter contains a highly conserved pentapeptide. In the close conformation as seen in the figure, the M2 helices are straight and cross over one another to form a helix bundle that seals the cytoplasmic phase of the pore. In the model shown, the channel opens when M2 helices bend at a specific point wherein a glycine residue is located. Now that we have seen how these prokaryotic potassium channels operate, we are in a better position to understand the structure and function of the more complex eukaryotic versions, which are thought to perform in a similar manner. Genes that encode in a variety of distinct voltage-gated potassium channels have been isolated and the molecular anatomy of their proteins scrutinized. These eukaryotic potassium channels contain membrane-associated helices named S1 to S6, while are shown two-dimensionally in this figure. These six helices can be grouped into two functional distinct domains. First one is a pure domain, in which has the same basic architecture as that of the entire bacterial channel illustrated in and contains this selectivity filter that promotes passage of potassium ions. The second one is a voltage sensor domain consisting of helices S1 to S4 that senses the voltage across the plasma membrane. So this figures shows the two alternate models of the three-dimensional structure of a single subunit of voltage gate potassium ion channel. So figure A is a classic model of the Drosophila channel, which is based on electrophysiological studies and structural analysis using non-crystallographic methods. The six membrane-associated helices are depicted as standard transmembrane segments. So figure B is the X-ray crystallographic model of a voltage potassium ion channel isolated from an archaebacterium that lives in 95 degrees Celsius ocean water. The protein displays the same physiologic features as eukaryotic uh, potassium channels, but is more easily crystallized. The S5 P S6 helices of the pure domain have the expected structure, but the S1 to S4 helices of the voltage sensing domain do not appear as conventional transmembrane helices. Rather than lying parallel to the helices of the pure domain, the S4 helices project from the edge of the protein into the lipid bilayer. So this figure shows the two alternate models of the movement of the S4 helix in orchestrating the opening of voltage-gated potassium ion channel. So in figure A, A conventional model based on type of structure shown in previous slide, in which the S4 helix moves charges across the membrane within the protected core of the protein, while the figure B, which is the new model based on the type of structure which is also shown in the previous slide, in which the like paddle through the lipid bilayer between the intracellular and extracellular surfaces. So figure A shows the three-dimensional model of an entire eukaryotic potassium ion channel showing the both membrane bond and cytoplasmic domains. Now, inactivation of channel activity occurs at the N-terminal portion of the polypeptide, which is called the inactivation peptide. So it fits into the cytoplasmic opening of the channel. Figure B shows schematic representation of view into a potassium ion channel perpendicular to the membrane from the cytoplasmic side, showing the channel in the closed, open, and inactive state. Substances always diffuse across a membrane from a region of higher concentration on one side to a region of lower concentration on the other side, but they do not always diffuse through the lipid bilayer or through the channel. In many cases, the diffusing substance first binds selectively to a membrane-spanning protein called facilitative transporter that facilitates the diffusion process. The binding of the solute to the facilitative transporter on one side of the membrane is thought to trigger a conformational changes in the protein, exposing the solute to the other surface of the membrane from where it can diffuse down its concentration gradient. An example of this mechanism is illustrated in this figure. Because they operate passively, that is without being coupled to an energy-releasing system, facilitated transporters can mediate the movement of solute equally well in both directions. The direction of net flux depends on the relative concentration of the substance on the two sides of the membrane. Facilitated diffusion, as this process is called, is similar in many ways to an enzyme catalyzed reaction. Like enzymes, facilitated transporters are specific for the molecules they transport, discriminating. Both enzymes and transporters exhibit saturation type kinetics as illustrated in this figure. Unlike ion channels, which can conduct millions of ions per second, most facilitated transporters can move only hundreds of thousands of solute molecules per second across the membrane. An example of facilitated diffusion is the glucose transporter. So, glucose is the body's primary source of direct energy, and most mammalian cells contain a membrane protein that facilitates the diffusion of glucose from the bloodstream into the cell, as seen in this illustration. So, a gradient favoring the continued diffusion of glucose into the cell is maintained by phosphorylating the sugar after it enters the cytoplasm, thus lowering the intracellular glucose concentration. So, humans have at least five related proteins that act as facilitating glucose transporters. Responsive cells share a common isoform of the facilitating glucose transporter. Now, when insulin levels are low, these cells contain relatively few glucose transporters on their surface. Instead, the transporters are present within the membranes of cytoplasmic vesicles. When insulin levels rise in response to elevated blood glucose levels, the hormone acts on target cells to stimulate the translocation of the vesicles from the cytoplasm to the cell surface, where the transporters become incorporated into the plasma membrane and transport glucose into the cell. The ability of the cell to generate such deep concentration gradients across its plasma membrane cannot occur by either simple or facilitated diffusion. Rather, these gradients must be generated by active transport. Like facilitated diffusion, active transport depends on integral membrane proteins that selectively bind a particular solute and move it across the membrane in a process driven by changes in the protein's conformation. Unlike facilitated diffusion, however, movement of a solute against the gradient requires the coupled input of energy. So the endergonic movement of ions or other solids across the membrane against a concentration gradient is coupled to an exergonic process such as the hydrolysis of ATP, the absorbance of light, the transport of electrons, or the flow of other substance down their gradients. Proteins that carry out active transport are often referred to as pump. In 1957, Jens Kuh, a Danish physiologist, discovered an ATP hydrolyzing enzymes in the nerve cells of the crab that was only active in the presence of both sodium ions and potassium ions. Sku proposed and correctly that these enzymes, which was responsible for ATP hydrolysis, was the same protein that was active in transporting the two ions. The enzymes was called sodium-potassium pump. So this figure shows the simplified schematic model of sodium ion and potassium ion ATPase transport cycle. So in number 1, shows the sodium ions bind to the protein on the inside of membrane. ATP is hydrolyzed and the phosphate is transferred to the protein. And then in number 2, the sodium ions change its conformation. And then in the number 3, because of the change in conformation, it allows the sodium ions to be expelled to the external space. Potassium ions then bind to the protein, and the phosphate group is subsequently lost, as being shown in number 4, which causes the protein to snap back to its original conformation, allowing the potassium ions to diffuse into the cell, as shown in figure number 5. So note that the actual sodium-potassium pump composed of two different membrane-spanning subunits: a larger subunit which carries out the transport activity, and a smaller subunit which functions primarily in the nucleation and assembly of the pump within the membrane. The calcium pump is present in the membranes of the endoplasmic reticulum, where it actively transfers calcium ions out of the cytosol into the lumen of the organelle. The transfer of calcium ions by the calcium ATPase is accompanied by large conformational changes, which are thought to couple the hydrolysis of ATP to the changes in axis and affinity of the ion binding sites. So, sodium and potassium pump is found only in the animal cells. This protein is thought to have evolved in primitive animals as the primary means to maintain cell volume and as the mechanism to generate steep gradients that play such a key role in the formation of impulse in nerve and muscle cells. Plant cells have hydrogen transporting P-type plasma membrane pump. In plants, this proton pump plays a key role in the secondary transport of solutes, in the control of cytosolic pH, and possibly in control of cell growth by means of acidification of the plant cell wall. In the resting state, the hydrogen-potassium ATPase molecules are present in the walls of cytoplasmic reticle. So, food entering the stomach triggers a cascade of hormone-stimulated reaction in the stomach wall, leading to the release of histamine. It binds to a receptor on the surface of the acid-secreting retial cells. Binding of histamine to its receptor stimulates a response that causes the hydrogen-potassium ATPase containing vesicles to fuse to the plasma membrane, forming dipoles on canaliculi. Once at the surface, the transport protein is activated, the pumps protons into the stomach cavity against the concentration gradient. So, heartburn drug Prilosec blocks acid filtration by directly inhibiting the hydrogen-potassium ATPase, whereas several other acid-blocking medications interfere with activation of the parietal cells. Acid-neutralizing medications provide basic amines that combine with the secreted retouch.